You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech Health Podcast. I have a returning guest, Dominic D'Agostino. He's a researcher and a professor with a diverse background in neuroscience, molecular pharmacology, nutrition, physiology. He's a PhD and a tenured associate professor in the Department of Molecular Pharmacology and Physiology at University of South Florida, Morsani College of Medicine. We've had him twice. This is the third time. He's a great guest. He's got a wealth of knowledge. He recently completed a mission kind of in partnership or collaboration with NASA, where they were under the sea and doing various experiments and, for a period of time to emulate what it would be like to travel in space. So that's my quick assessment of it, but I'll have a much better way to tell you about it. So, Dom, thanks for coming back. How you doing?
I'm doing well. Thanks, Richard, for having me.
Yeah. So tell me, what was this NASA mission, and what was the goal of it, and what was it like?
Sure. So NASA does a lot of training to prepare their astronauts for deep space exploration, or even not even deep space, just to the space station, for example. And they have a number of what they call space analogs, and there are a variety of different analogs that researchers take part in. And these analogs, the main thing is to vet out technologies and concepts that can then move to space. And most of the analogs, all of them actually, excluding the one that I was part of and my wife was recently part of too, use just everyday people. But the NASA Extreme Environment Mission Operation analog, and that we just completed number 23 expedition, trains astronauts, and it's part of their training for space exploration. And within the analog, it's undersea. So when you're in the water, that simulates neutral buoyancy. So, or microgravity. You could be weighted down for the surface of the lunar surface or the surface of Mars. And typically you're inside a habitat, and they try to mimic particular situations, whether it be lunar or Mars exploration. And inside the habitat, it's dry. So we're living underwater. The aquanauts, when you live underwater for more than 24 hours, you become an aquanaut, and 60 or more astronauts are aquanauts. And so when you live underwater for more than 24 hours, you become an aquanaut. And during that time in the habitat and outside the habitat, when you're walking on the floor of the Atlantic, which is where the habitat is based, you do a variety of objectives that are tightly correlated with space missions. And that can be, tools, concepts, different devices that NASA has. And they do this in partnership with the European Space Agency. So ESA has a bunch of objectives too. So it becomes a really intense mission that's really designed to mimic the space environment.
Yeah, so I mean, outside of the habitat, you're in a suit, like a diving suit, and you're in the water, so I can see it would emulate microgravity. Yeah. But inside the habitat, is there any way for them to adjust the condition so that it somehow resembles lesser gravity, or is it just you can only emulate the spacewalk part of it?
Yeah, that's a good question. So the only thing that's really not mimicked in the habitat would be gravity, right? So, and perhaps in future long duration space flights, maybe they can mimic gravity, like Space Odyssey, 2001 Space Odyssey, where they have essentially a centrifuge or something like that, and that lack of gravity really contributes to many problems. But there's also space radiation. But believe it or not, the biggest challenge that deep space exploration presents is really living in that confined habitat for long duration. So a lot of the objectives will center around things like team cognition, psychological health, sleep, being able to perform a variety of scientific experiments and tasks in a habitat, an enclosed habitat that's very small and with limited space. So it becomes sort of, we test a variety of procedures, from using various software that tells us when we have to do things and particular, sequence, and doing things, and how one crew member's task will be sort of staggered or coordinated with another crew member's task. We use something called Playbook, and for that, and they try to mimic what's kind of going on the space station, and we'll use devices to measure, the microbiome of the habitat, the microbiome of our skin, and we can sequence the DNA with a mini DNA analyzer that I used on my mission. Actually, one of the first tasks that I did was to sequence, the microbiome of my skin and the microbiome of the habitat. So the undersea habitat is also under hyperbaric pressure. It's 3 times the pressure of a normal habitat. So that means the air is thicker, and that's exposing us to a physiological stress. So we do a variety of things to monitor the physiology, because we're breathing, 3 times the amount of oxygen and nitrogen and things like that. But yeah, we are lacking that, gravity. So the gravity is essentially the same in the habitat. But when we go out during the day, we go out and do what's called EVAs, extravehicular activities. And those activities can be things like testing a device to rescue an incapacitated astronaut. It could be using a specific drill that can enable us to get a sample of a rock or a coral. Part of what we do, we work with the Coral Reef Foundation to take samples of coral, ID them genetically even, and then put them on cards, and then we grow them in coral nurseries so we can propagate different species that are endangered, like Orbicella and Siderastria, I think, are 2 of the species that we would try to collect and propagate.
So you're doing some useful work, not just testing stuff?
Yeah. So what NASA does is they partner with different organizations, for example, the Coral Reef Foundation, and our actual mission objectives will support other objectives, environmental objectives, engineering objectives. My wife was on MIMO 23, and she partners with National Geographic, and they have an ROV device, basically an underwater drone kind of device. So one of her projects was to use this underwater drone at nighttime, and she would be dry inside the habitat, but she would be operating it. The ROV would be collecting things like plankton samples. Or actually been able to, you can go at nighttime with the lights and everything and just observe the habitat, look at various, electrical lines or or various parts of the habitat to check it, to make sure things are working properly. So she partners with National Geographic to do some observational work with that. And and we have various devices from a wide range of different companies that, are hoping to get their technology into space.
All right, so some questions about your living conditions. How long were you down there?
Yeah, my mission was 10 days living in saturation. And that includes, at the end of it, it was about 16 hours of decompression. So a staged decompression to come out of that environment. So yeah, living in 10 days in that environment with a very tight schedule from morning till night, doing experiments.
Well, that's probably a good thing. Otherwise you might go crazy, or, the people that you're there with in close proximity, you might, get into a fight or annoy the shit out of each other if you weren't busy.
Yeah, well, they love to study that, right? Because like I mentioned, part of the biggest challenge is really being able to select a team that can work together and have that, team dynamic or efficient team cognition where everybody's kind of on the same page. And things can break down quick under certain stressful situations. And they try to, you get hit with a lot of stressful situations if something goes wrong, if, a device breaks and then it backs up the timeline to where another person can't use it. The psychologists are kind of analyzing how that impacting the psychology of the crew. And to assess this, there are a wide variety of questionnaires that we'll do in written format and online format. And then there's also a number of sort of iPad, devices that we use to assess psychological health, even things like reaction time or decision making. One of them is an NIH toolbox, and another is called Joggle. And Joggle is a software sort of psychology testing device that's actually used on the space station. So we try to mimic and use some of the tools they have down here, and that assesses the crew's psychological well-being, because being confined into a small environment, if you have 6 people in a space, that's kind of correlates to like a 30-foot RV, like camping RV, and they have to live down there and they're tasked with a lot of work to do. But believe it or not, my crew, my commander was Shell Lindgren, and he's a seasoned NASA astronaut, and also had Pedro Decu, and he was a seasoned European Space Agency astronaut. These astronauts, they're obviously they are selected for this type of occupation. So they are, many people apply to be an astronaut, and only a few people make that cut. So they have to pass a number of psychological tests and criteria to even be an astronaut. So I knew going into this that I was going to be among people who have, been in extremely confined environments with crews and being able to efficiently in those environments. And I learned tremendously just by watching them and being among them. Yeah, I really had to kind of step up my game, right? Because
any life skills you developed from being around these people? Or I was going to make a joke that it's a good place to do marriage counseling, or a bad place.
The life skills are really probably the biggest take home was really seeing how professional astronauts are and being able to, the commander, for example, taking that leadership role and holding himself accountable to things that go wrong, even if it wasn't his fault, because he's the commander of the crew, right? And being able to make important decisions if something's not working, to completely abandon that particular task and reallocate time to things that should take priority, especially in regards to things that will give things that'll keep us safe, for one thing, but things that'll also give the greatest scientific kind of return. And And, the space station is really just like a scientific laboratory. I would say 50% or more of what's done on the space station is really to do science. And I would say our mission too was probably about 50%. It kind of mimics that to do science, and maybe lumped in with that for another category is to vet out different technologies. And that could be various apps, training, like physical training devices, and and a wide variety of things for monitoring health and monitoring sleep and monitoring our stress levels.
Well, let's get into all the science part of it. So you were at 3 atmospheres of pressure for 10 days, essentially?
Yeah. Yep. We were,
Wow.
Aquarius is 62 feet below the sea surface. And unlike a submarine, submarines can dive down, obviously far deeper than that, but a submarine stays at 1 atmosphere, right? So the Aquarius habitat, research habitat, it's like taking a cup of water or taking a cup of air and turning the cup upside down and then pushing that cup into a pool and then going all the way down to 62 feet, right? That cup will essentially be compressed to 3 atmospheres absolute. 3 total atmospheres, where you're at 1 atmosphere now at your desk or wherever you're at, unless you're, in the mountains somewhere, then that would be even less. So you're living, and that pressure, that level of pressure is definitely altering our physiology. And when you scuba dive, you go down to, similar depth, 62 feet for recreational scuba diving. And then you have to come up at a predetermined time point so you don't have what's called, so you don't get decompression sickness, right? But with saturation diving and living in saturation, our bodies are saturated with the oxygen and the nitrogen. And it's the nitrogen that can cause a lot of problems if we try to come up to the surface. So after living down there for a few days, if someone was to get an infection or break a bone or something like that, and if they were to shoot up to the top of the surface, they would die a very painful death if they were not rapidly put into a recompression chamber or hyperbaric chamber, because you need
the nitrogen would come out of their blood and would cause an embolism or a blockage.
Exactly. Yeah. So you would have decompression sickness, and maybe more commonly or also known as DENS. And when you open a bottle of soda and you see, a carbonated beverage and you see the bubbles coming out, that's essentially what's happening in your arteries and your blood vessels and your tissues. And those bubbles can impede blood flow and cause a lot of problems. So there are in scuba diving, there are protocols that you need to adhere to. And as in military diving, and this is part of the training, we learn about that as part of being a saturation diver. And there's a fairly complex stage decompression to come out of that environment. So my commander, Shell, actually posted on Twitter, it took him far less time to return to Earth from the space station than it did to return to the surface of Earth from MIMO, right? It took like 17 hours for MIMO, and it takes about 5 hours, give or take, for astronauts to get back to Earth from the space station. So, so it takes actually longer, and a lot of the astronauts that I've talked to who have been aquanauts in that environment, and the commander for the last mission that my wife served on, Samantha Cristoforetti, the Italian European Space Agency astronaut, said that it really did. She was in space for 200 consecutive days. She said it really did simulate, sort of from an operational perspective, living and working in space. And my commander said the same thing, as did the other astronauts that were part of the crew.
So I've heard of, hyperbaric oxygen therapy, but that's, for an hour or 2 hours, 90 minutes. What physiologically have you identified that it does to you being at 3 atmospheres of pressure for 10 days?
Yeah, that's a good question. So I would have to say for humans that it wasn't even known really in the 1940s. I guess in the 1950s with the Sea Lab missions, did it become known that humans could actually live in a saturation environment for weeks or even months on end. Sea Lab 1, 2, and 3 kind of researched this, and so it was like in the mid 50s. Prior to that, it was thought that humans couldn't even live or work in these environments for prolonged periods of time without having to desaturate or decompress. So there are a number of things that happen. One is that the partial pressure of oxygen usually goes up, and that can increase reactive oxygen species in the body and increase inflammatory markers. There are changes in renal physiology. There's a mild diuretic effect that happens in that environment. Acutely, some hormones start to change, like cortisol goes up. But what's most striking is that humans are essentially non-compressible, right? Because fluids and solids are not compressible, but air is, right? So the only really air-filled spaces we have are sort of our lungs and our esophagus and everything. And then the air going in there is, at a higher degree of compression, but it's not really compressing us per se, because we're fluid. We're basically like water sacs, right? I mean, it's like proteins, electrolytes, and H2O. So we're essentially non-compressible. So the remarkable thing is that living in that extreme environment of 3 times the amount of pressure does not crush you in any way. It's just, it does have some physiological effects and is a stress to the body, probably due to the partial pressures of gases that we're breathing. But if that's mimicked to simulate the partial pressures or the oxygen concentration, I would say, of a normobaric environment, much of the effects can be mitigated if we titrate oxygen to the right level. Too little oxygen, then you become hypoxic. Too much oxygen, and that's hyperoxic, and your body will proportionally make more reactive oxygen species in response to a higher partial pressure of oxygen. So if we were to breathe, and we do before decompressing, we breathe like 100% oxygen for an hour as part of the initiation of our decompression. Now, if we were to breathe, if we breathe 100% oxygen in that environment for longer, we would have oxygen toxicity fairly rapidly, and which could be seizures, and then later pulmonary oxygen toxicity, which can damage our lungs. So
what was the mixture of air? What the mixture of air we breathe that normally is,
yes,
I guess approximately 19% oxygen, et cetera. What was it down there? Did they titrate it differently?
It was, we were getting air essentially from the environment that are sort of pumped into tanks, and then those tanks are sort of connected, and there's positive flow of air coming in, and then air is kind of leaking out, so it's constantly being flushed, and then the CO2 is being scrubbed. So it's essentially 3 atmospheres absolute of oxygen. So, on an order of 3 times higher than what you would be breathing. And nitrogen too would be higher, and nitrogen's inert. Well, nitrogen is inert at that pressure, but if we were to go down to 100 feet of seawater or lower, 200 or 300, nitrogen has a narcotic effect. So, and I believe it has a very mild narcotic effect at, that level at 62 feet. The biggest issue is in space travel too, and this also relates to the space station, is that the concentration of carbon dioxide is 10 to 20 times higher in this environment and is very similar to the space station. And that's the biggest concern from a physiological point of view. is the high concentration of carbon dioxide that continuously needs to be scrubbed out.
Well, and so is it kept, the best they can do is keep it at 10 to 20 times higher, or are they able to keep it at what it's like at the surface, or they can't? They just physically can't?
Well, yeah, they do. They have systems in place, and the space station has two systems in place that are very complex systems to scrub out the CO2, but they only operate one of the systems. So if one breaks, they can operate the other. So one of the questions is, should they run both CO2 scrubber systems at the same time to decrease the partial pressure of CO2 from, for example, 2,000 parts per million down to 500 parts per million, or something that would be closer. Humans breathe about 400 parts per million or thereabouts. So it gets around 3,000 parts per million. So it gets about 10 times higher. And that's about what it stays in the MIMO habitat. And in a couple cases, it jumped up to about 10,000 parts per million. And like the red buzzer went off, and then we had to exchange the scrubber material that scrubs out the CO2. So it's essentially sodium hydroxide or lithium hydroxide. There's various scrubber materials. So we basically have to pull the scrubbers out and dump in new material that helps pull the CO2 out of the air as the ventilation system runs it through the system. And the monitor will tell you essentially when that needs to be done, because when you change the scrubber material, you see a very refreshing drop in CO2. And then as time goes on and the scrubber material becomes sort of exhausted or consumed by consuming CO2, or you get more crew members come in the habitat, more activity, or you're doing physical activity, say you're exercising in the habitat, you'll see that reflect in the CO2 level. And by far the most, the biggest problem that presents itself in a confined environment, a space environment or undersea environment, has not necessarily been oxygen. That's a concern, but CO2 has been a far bigger concern. That's reflected in the movie, in Apollo 13, right, where they had to essentially build a CO2 scrubber to stay alive on that movie. Or in that mission. So CO2 is a big problem. And I know Scott Kelly, who stayed in space for a year or more, has been a very vocal advocate of the CO2 problem in space, and has really advised NASA. In his book, I think he mentions it 58 times throughout his book called Endurance, which is a fantastic book for your listeners. Really, his message was that the CO2 problem is a very big problem that needs to be mitigated. And I know as a research scientist that elevating CO2 up to 2,000 to 3,000 parts per million, which would reflect the International Space Station or the MIMO habitat, starts to impact things like inflammatory pathways in the body, and even the intestinal mucosa. So the tight junctions that hold the epithelial cells together start to be impacted by CO2 and are sensitive to that. So your gut actually becomes more leaky, and your gut permeability can really impact your overall immune health. Most of our immune system is actually in our gut. So when that's compromised, it lets the external environment enter into the blood, and there are proteins and molecules that can wreak havoc when we have a leaky gut. So, as a research scientist,
there's so much
to
talk about here.
Yeah. Well, yeah. So the CO2 is probably one of the bigger things that we've been monitoring, and I've been making measurements on myself, and we have physiological measurements and sleep measurements. But I think in the future, in space analog missions, we'll probably want to direct more resources and funding to really understanding this more, because there's a lot of things that happen and we don't understand why hypercapnia, which is high CO2, is causing these problems.
Well, yeah, just thinking about it. So oxygen is at a higher pressure, so it's being driven into there's more of a driving force, I guess, to go into your body, into your tissues. But then the CO2 concentration being higher, there would be less of a driving force, I would think, to expel it, through your lungs. So I don't know how that would change the composition, the relative compositions of the two and tissue saturation in your body. What have you observed?
Has some interesting physiological effects. It's a vasodilator, and because it dilates blood vessels, it can actually increase blood flow, maybe even brain blood flow. And a lot of astronauts report getting headaches. So, and they report that those headaches go away on the space station if the CO2 levels drop. And that could be by, fixing the CO2 monitor or CO2 scrubber, or if there's a bunch of crew members that leave the space station and there's less crew inside the space station, the CO2 levels will drop and they will be able to feel that. And I know astronauts like Scott Kelly are very sensitive, and other astronauts like Samantha Cristoforetti said it was sort of like a problem, but not like a problem that really impaired her performance, she felt personally. Although she did say when the scrubber was fixed, it was like walking through a forest, like the air was very fresh, and that she could feel that. But for other people, it becomes like a really big problem. The dynamics change at the level of the lung, but the biggest effect CO2 has is that it's a potent vasodilator, and it can also CO2 can react with various redox biochemistry pathways to exacerbate or increase the formation of certain reactive oxygen species. And that's something that we actually study in the lab. So you can actually get higher oxidative stress with the same amount of oxygen if it's at a higher partial pressure of CO2. And we are looking at this in the brain, but I think it can, it's ubiquitous throughout the body. So you're getting a greater oxidative hit. So I think it might be important to understand how astronauts or people in extreme environments can augment their antioxidant defenses. And that could be by stimulating our own natural endogenous antioxidant mechanisms. And ketones, we feel, do that. Ketones are epigenetic regulators that increase the expression of enzymes and systems in the body that can allow us to cope with high levels of oxidative stress. And ketones can be elevated with the ketogenic diet or exogenous ketones or fasting or a combination of those three things. And, it may be more important to supplement with antioxidants. We're pretty sure it's maybe more important to supplement with antioxidants when we're in these extreme environments. And that has been sort of an area of ongoing research, even in past MIMO missions, looking at oxidative markers in the blood and inflammatory markers. Inflammation can increase as a consequence of high levels of oxidative stress. So I think it's an ongoing area of research.
Well, what's the constraint? It's just not energy efficient to run the scrubbers hard enough, or they want to see, okay, they figure, all right, well, 2,000 parts per million is tolerable. We're not going to make any effort to get it lower than that. I mean, what's the constraint here? Why can't they make the air quality better?
Yeah, that's a really good point. So it's, let me see how to put it in terms. So you could probably, it takes a certain amount of effort to get the CO2 level concentration from like 20,000 parts per million, right? Which would really start to impact and make people sick, down to 2,000 parts per million. So it takes, a certain amount of effort, right? It'll take an equal amount of effort to bring that 2,000 parts per million down to like 500 parts per million. You know what I mean? So it's relatively easy to put filtration systems in place to move the air, to make it tolerable and within the limitations of federal agencies for safety, but the amount of the energetic cost, the resources, and the maintenance associated with bringing that 2,000 parts per million CO2 level down to what is ambient level of CO2, like 300 or 400 parts per million. That's a pretty big challenge. So I think what has not been accepted, although the undersea medicine community, the Navy, is probably more advanced in their understanding of the CO2 consequences on the body than NASA is. And I know those two agencies are talking about mitigation strategies for CO2. And I think there's a lot of technologies that are being developed, like in submarines, for example, that can be applied to space habitat. But they have a very complicated part of the problem is that the system that's on the space station now is incredibly complicated, and it's difficult to fix that system when it goes down. And it takes an entire day really to basically take that system apart and fix it. And there's two of them. And if they both break, then there's a really big problem, right? But I think technology and systems will evolve that will allow these habitats to basically come fairly close to ambient conditions. But we're not at that level right now.
Well, what about plants? Could you have a whole ceiling composed of lichen or moss or something, and would that help out, or are plants not nearly efficient enough to scrub the air?
Well, yeah, they do contribute. So plants that undergo photosynthesis and that grow, that the photosynthesis and that carbon that is essentially, the growing plant that was once CO2, right? So that's why things like, believe it or not, corn and grasses and I guess the rainforest, the things that proliferate from a photosynthetic point of view, and light drives the photosynthetic process, really captures a lot of carbon. So that's one strategy, but it's like, in regards to metabolic CO2 production, it's not a whole lot. If you have like a base on Mars or a base on the lunar surface, it would be advantageous to have a wide range of plants growing that you could optimally use for food that would be capturing that carbon. And I think, probably you can even genetically modify various types of food to grow faster and to suck up more, essentially, carbon. So I think that's
I just wonder if, is there a calculation of, for one respiring human of a certain weight, how many plants are needed to offset that person's carbon dioxide production by, 50% or 100%?
Yeah, and we know that, and we think like something like a biodome, in those experiments where CO2 was monitored, the CO2 burden of a respiring human will far exceed the CO2 capturing capacity of plants in a confined habitat like the space station. But if you had a large area like a biodome or something that's on the surface of a planet where you have more room to create, then you could have a wide variety of plants that could probably deal with all the CO2 burden if it wasn't heavily populated by humans. So that's easy to do and has been studied in different ways. But not for a confined space, not for a space capsule or not for a space station, but that's a big project, and people are working on that project when it comes to the lunar surface or the Mars surface, is creating sort of these natural CO2 sinks that are really would not only suck up CO2 but be producing food, and that's what plants do really well, and designing agricultural strategies to do that. and using things like aquaponics is another strategy to do that.
So what kind of experiments did you do on yourself, and what did you notice? How did your body react? You mentioned cortisol being elevated. Microbiome you sampled. Did this put a selection pressure on the microbiome of your skin or your mouth or other places you sampled from? And did you notice changes? I mean, what else did you see happening?
Yeah, the microbiome samples that we have for gut microbiome still have to be analyzed. So we've just collected another crew sample from MIMO 23. I was on MIMO 22. And, over the course of a year, the cost of analyzing these samples just keeps dropping. And the accuracy and the data you get back is greater. So waiting to analyze that has not really hurt us in any way, especially from a financial perspective. We did the microbiome of our skin, which we sort of sequenced it using the mini DNA analyzer, was the results suggest that our skin microbiome starts to reflect the microbiome that is the bugs that are present in the ocean. So those bugs tend to live on us even after we've showered and rinsed off and things like that. They tend to live and grow on our skin when you're living in the undersea environment. That kind of makes sense, right? But so they're not. We don't think they're hurting us in any way, but some people in that habitat, living in that habitat, broke out in hives or had strange rashes, and we never really determined what was causing those rashes. I didn't have any problems, so maybe that's evidence that ketones can be protective against changes in the microenvironment or the skin microbiome. So, but
you work with ketones a lot. Did you? Were you on a ketogenic diet? Were the other people in the environment on the same diet or different diets?
So the objective was to really understand how to not change any variables in regards to diet. Since I was already on a ketogenic diet, I was able to. I continued with a ketogenic diet, and I'm sort of an outlier in that way because it is interesting to compare, and I can't talk about it until it gets published, to compare my physiological and even psychological response to the extreme environment compared to others who were not in a state of nutritional ketosis. Like how did certain parameters change if you're in ketosis relative to people who were not on a ketogenic diet and maybe didn't have that. So we have data from 10 people now, and they were all on what would be equivalent to their standard diet. So the food down there sort of reflected what they would eat normally, which was not a ketogenic diet. But my food, since my normal diet is ketogenic, I stayed in a state of ketosis throughout the entire mission. And I even recorded when I came out of an extravehicular activity, an EVA that was like a little over 5 hours, I believe, my blood glucose level was down to like 2.3 millimolar, and my ketone levels were up in the 6 millimolar range or more or higher, like 6.6. So my ketone levels were 3 times higher than my glucose. So what essentially that means, and our body can use both of those fuels fairly readily, that my brain, the majority of my brain energy was being derived from ketone bodies. And I did feel really good coming out of it. And I had to measure again to make sure those numbers were correct. But yeah, that was probably a consequence of being on the ketogenic diet and then working in the water. Water can pull heat away from your body much faster, even with a wetsuit. So even though the water was pretty warm, because it was, in the Keys in June, by the time I got out, I was in a state of mild hypothermia, even though you don't really feel it. You start shaking a little bit uncontrollably, kind of like a little tremor in your hands. You can kind of feel it, but it reflects metabolically as an increase in metabolism and probably things like catecholamines and stuff, which I measured and were elevated. So, and we also have sleep data too, and HRV data, which kind of can give some, and we're still analyzing that data and putting the new group of crew and analyzing that, and will reflect sort of the balance between sympathetic and parasympathetic tone and how that changes or how that's altered.
Yeah,
before.
It sounds like there's so much data that you could pass it out to, I don't know, 4 or 5 grad students and have them all work up papers on all the things that you recorded just on yourself.
Absolutely. We could probably take a year off and just analyze and write papers. But we have to continue sort of chugging along and analyzing in little bits and pieces while we maintain sort of the lab research, the active projects we have going on. And the data is very sensitive because we got approval through not only, our institution, but also NASA and ESA. And when you're dealing with data from astronauts, there's an extremely high level of oversight. So anyone who sees the data, touches data, or is a part of an analysis needs to be on the IRB protocol, like the ethics committee protocol. Even things like the microbiome, you have to go through a lot of extra paperwork because you're kind of hold the astronaut's DNA, right? Because cells from the fecal sample could be from the actual astronaut, and you could basically derive who that astronaut was, even though all the data is de-identified. And you could potentially come up, and show that this DNA shows some kind of SNP that would suggest that if they went into deep space, they would be compromised by the radiation in that space. Like these are the sort of things that NASA sort of are concerned about, being able to, that data from astronauts could be, put into the public domain, and that could impact their future potential for being on a mission. So there's a lot to consider when dealing with this, and that could be, genetic data is a very high concern, but it could be physiological data, biochemical data, psychological data. So all this stuff, we need to move very slowly and methodically as we analyze this and think about writing it up, because the implications, because you can go online and Wikipedia and look up MIMO and see who the crew members were. So you could say, oh, well, this person does not react, someone in the crew did not react very well to this psychological test or something like that. So, or didn't react well to that intervention. So there's a lot of, and these are things I didn't really consider or kind of know about getting into this research, but we have to kind of work closely and work very thoughtfully as we put this data together, that it doesn't make any suggestions that key members of the crew may not be adaptive to these extreme environments. So we have to consider the results as we move forward. But we like to see big changes, right? Because these extreme environments produce big changes. And our overall, what I've spent my career on is developing sort of protocols that are countermeasures to these extreme environments that would allow us to have physical, psychological, and neurological resilience in these extreme environments. And that means changes in the partial pressure of oxygen and CO2 and just, things that are associated with extreme environments. It's like we want to preserve our physical capabilities to enhance mission success and safety.
Well, you could look at, I mean, it's too much of a stretch, but I mean, you could look at aging, or you could look at a whole host of conditions that someone lives in as extreme environments of their own, having cancer or whatever it may be. So, the work you're doing,
for sure. Yep.
I'm sure it applies way beyond just, literally these kind of extreme environments.
It does. And it's a little scary, but these environments where there's a high partial pressure of oxygen, in theory could be accelerating the aging process. And many people, there's many that the redox theory of aging would suggest that, but the data so far really does not suggest that. And even Scott Kelly's data, I mean, his twin, who stayed on Earth, Mark, did not, really suggest that being in space environment where you're exposed to considerably more radiation and factors that should be speeding the aging process, that wasn't clear that it is accelerating processes that are associated with aging. And I think what's more important, and I think will be a key area of research, is the epigenetic effect of things like exercise, nutrition, and even psychological well-being, and how, from an epigenetic perspective, not our given sort of somatic DNA blueprint, but the expression of those genes and how we can manipulate, suppress, or increase the expression of certain genes that will give us resilience, protection, and optimize us in these extreme environments. That's becoming progressively a topic of greater interest in funding agencies like DARPA, DoD. I know the Office of Navy Research is really interested in epigenetic modification, and whether that be a drug, nutrition, or a nutritional supplement to augment that.
Well, I think with the Kelly twins, it was observed that hundreds and hundreds of, maybe even thousands of changes occurred epigenetically and genetically, right? Or epigenetically, I mean.
Yeah.
But then reversed?
Yeah. The data is still being sort of analyzed and interpreted in different ways. But I think there was, and it got taken out of context in different ways. But I think the take-home message is that, these extreme environments do impact us from the standpoint that it impacts the expression of certain genes, and does not necessarily fundamentally change our genetic blueprint in a big way, but it's more likely to change the expression of genes. And I think really that should be the focus, because you could take many different stressors, here, exercise, a drug, a food, high sugar, high fat, low carb, many different things can alter, probably in a more dramatic way, these epigenetic modifiers that can influence ultimately our blueprint over time. But I think
what's your overall assessment of our likelihood of colonizing Mars or colonizing the moon? Now that you've been in an extreme environment and you've been around some of the technology involved and the challenges, do you think it's far harder than we think, easier than we think? What are your general thoughts there?
Well, I think a lot of steps are being, taken to colonize the lunar surface first, right, in a way that's safer and not as restrictive. The Apollo mission was an incredible. I keep revisiting like books and movies and stories associated with the Apollo mission. It's just amazing that we were able to pull that off. And I think the next challenge is to really, make it safe and make it productive to land on the lunar surface, and to set up systems and strategies in place as a launching pad, ultimately a learning place and a launching pad for Mars exploration. So I think it's, NASA has been very cautious. A lot of people ask, like, why haven't we been to the moon, in 50 years? And I think there's a lot of good reasons for that. That the research and the time spent through all the shuttle missions and, on the space station too, the research that was done there, that research is going into putting together systems in place to make it safer and more efficient and more cost effective to explore the moon and to ultimately go to Mars, which the dates keeps getting pushed back. But I think that's for a good reason. I think we need to the whole sort of political environment could change pretty fast if there's another dramatic, catastrophic incident if we go to the moon. So I think it's been delayed, for good reasons. And I think the science that the essential science that was done on the International Space Station, which is a floating research station, that science will be put into very good use. And it's just, a matter of time. But it really does come down to research dollars, federal research dollars, and private industry to drive this next phase of exploration.
So now that you learn things, being in this environment for 10 days, is it changing the focus or direction of your research? Or are you still continuing as you were? Or do you have new insights that now you want to change things up?
Yeah, well, I spent many years as a basic research scientist studying like cellular and molecular mechanisms of extreme environments. And then, as my research program advanced, we were able to move those things into a wide range of animal models. So now we're doing mice, rats, and even pig studies. And now I'm gradually shifting work from the basic science proof of concept to the human implementation of these strategies. And that could be in a university setting or in an operational setting, like on an MIMO mission. So I would like to allocate more time and effort and resources to directing our science into more human operational conditions. So I see that as a big part of our growing research objectives, is to allocate more time, energy, and funding into to actual, human-centered operational type missions, where we're vetting out different procedures, technologies, and ketone technologies, and ways to enhance our metabolic physiology to make us more resilient in these extreme environments. And there's many ways to do it. And I think we just have to move methodically and vet out what works best in all these different model systems, and then apply what we conceive as the optimal approach in these training missions for NASA, and ultimately maybe take some of these technologies to space.
Were you in this in MIMO with your wife, or was she there at a different time than you?
Yeah, my mission was actually in 2017, so it's been a while, but we still have data that we're analyzing. And my wife's mission in 2018, the mission was canceled because the hurricane Irma really damaged the habitat. So my wife's mission was an all-female crew, and that was over the summer. And she spent 9 days. And there was a delay going down on the first day, but she was spent 9 days in the habitat. So we essentially repeated many, if not all, of the experiments I did on my mission on MIMO 22 on her mission. And it's great that we had a female crew, so now we have a good balance of male and female data, and we can look at the difference too. An important difference is to understand how males and females may respond differently in these environments.
Well, it was a joke if you were down there at the same time she was. It would be a test of your relationship. Right?
Yeah. Well, I did when I was down there for my mission. I actually had my birthday down there, and she came down as a support diver and delivered like a birthday gift to me. And so I returned the favor, although she didn't have a birthday down there. I delivered, a small gift to her when I function as a support diver, and I also function as a mission control sort of research coordinator on mainland. And when I wasn't on mainland, I did some support diving for the mission. So, and she was part of my support diving team, when I was down on mission 22. So it's pretty good that we're very fortunate that we were able to both participate in this, and working with NASA has been probably one of the most incredible experiences of our lives, just being selected for this type of research and to get our science on these missions has really been amazing to us, and just working with them and seeing how professional they are. So we look forward to partnering with them in the future and hopefully doing more missions with them. And even if we can't be crew members, we can get our science on the mission so we can continue to collect more data and refine sort of our understanding of how these extreme environments impact, things like microbiome, sleep, stress, psychological parameters, and then further sort of develop and refine our mitigation strategies to optimize our health and our performance in these environments.
So, all right. So when do you think some of your data will be coming out, and where can people find it when it does come out?
Yeah, well, we'll definitely be posting it on my website, ketonutrition.org. But we will first, we'll put together the manuscripts for publication in peer-reviewed journals, which would be found on PubMed. And prior to that, though, we'll probably start presenting, now that we have 2 groups of data and we have a high enough sample size to get some statistics going, we'll probably start putting together the preliminary reports and presenting at some of the annual conferences like undersea UHMS, Undersea Hyperbaric Medicine Society, and also the Aerospace Medicine Society meetings, or where we have presented historically in the past. And then that helps us get feedback on our actual study in sort of preliminary format. And then we use that feedback to help further refine the overall study and manuscript for publication. So yeah, I travel around and give talks on the meeting and stuff too, but just really haven't not put it together yet because there's a lot of deep analysis going into understanding these things. Just doing the HRV data alone, we have a massive amount of it. So we have another group of people just working on that project alone. And then the microbiome project, as I mentioned, we're negotiating prices on the best way to analyze this so we can get some fruitful data from the samples that we have.
Well, very good. Well done. It's always great to have you. You generate more data than anyone I know, and you get to have the fun and other people analyze it. Yeah, I guess it could be argued.
I have to attribute that to my team, though. Yeah, so I mean, it's good to be it's nice to be kind of like the head person, the go-to person, but it's really our whole research team, and that includes many people that like Andrew Gutnick too, who was an amazing asset for my mission. He really ran the sleep studies and the body composition studies and stuff too. So yeah, I'm very fortunate to have an incredible crew of researchers working under me.
Yeah, that's fantastic. Well, Dom, thanks again for coming. Third time's the charm. I really appreciate you being here.
I appreciate the opportunity. Thank you, Richard.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.